Hello, everybody. Welcome back to lecture. So we are on chapter 6. This chapter is kind of like a split topic. They kind of cram two topics together. So the first half of the chapter is dealing with requirements for bacterial growth, kind of metabolism, nutrient requirements, oxygen, energy, those types of things. And then the second half is dealing with actually growing bacteria in a lab. So you're going to be experiencing that with your lab activities. So we're going to start off with the chemical and energy requirements for bacterial growth. And these requirements help to classify some of these organisms. All right, so in this table, it is breaking up the energy and chemical requirements. Now, when we talk about the energy, it is what is the source of energy to drive ATP synthase, which is the production of energy. So it's kind of a interesting concept. So for example, our two energy sources are light and chemical compounds. So going back to chapter 5, which we just got done, we just finished, we saw different types of phosphorylation, substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. So when we are talking about the phosphorylation, that is our energy. We're making ATP by adding phosphate groups. In oxidative phosphorylation, it was the breakdown of chemicals, right? So glucose is what we started with. We broke it down systematically, taking the electrons, making a little bit of ATP via substrate level phosphorylation, but really it was those electrons that we stripped away from the carbon molecules that drove the electron transport chain, that made the hydrogen ion gradient, that drove ATP synthase, that got you ATP. Those are the chemicals. That's where the energy comes from, breaking down those chemicals. When we looked at photosynthesis and it was photophosphorylation, it was the light energy. It wasn't breaking down chemicals. It was the energy in the light that excited those electrons, allowing them to go down an electron transport chain, pumping protons to drive ATP synthase. So when we talk about an energy source, that's what we're talking about. Where does the energy come from? Basically to drive the electron transport chain. Does it come from stripping those electrons away from chemicals, which would be a chemical energy source, or light exciting the electrons in the chloroplast or from the water in photosynthesis and that photophosphorylation. So those are our energy sources. Okay. Then when we take a look at our carbon source, the carbon source is where does the carbon come from that we need to make new molecules, like in those anabolic pathways? If that carbon comes from carbon dioxide, like in a photosynthetic organism, it goes to that Calvin-Benson cycle. The CO2 comes in, gets hooked together to make glucose. So that is where we could get carbon from, this inorganic source of carbon dioxide. In other anabolic pathways, that carbon comes from organic molecules like glucose or proteins or fats or nucleic acids or whatever. So those carbons are not carbon dioxide. It's basically other organic molecules besides CO2. All right, so that's where those terms or what we're talking about, the energy source versus the carbon source. So then we have these, let's see, root words. Photo for light energy, chemo for chemical energy, auto for carbon dioxide carbon, and hetero for organic sources of carbon. So then we can mix and match depending on what the organism does. Let's take a look at what we are, humans and a lot of other organisms. We are chemo heterotrophs. And troph is just a root that kind of means like nutrient level or requirement. So chemo heterotrophs, that's a clue. That tells you these organisms get their energy from breaking down molecules. They strip their electrons away from chemicals. That's the chemo part. And then the hetero part lets you know that the carbon that they use to build more of their own molecules, like that what they need to use to make amino acids and carbohydrates and nucleic acids and fatty acids. They get that from consuming organic compounds. So we are chemoheterotrophs, right? We are most animals. And a lot of bacteria and protozoa and fungi and many bacteria fall into this same category. Some fermentation like yeast and some other types of bacteria, but these are chemoheterotrophs, okay? Now let's take a look at the other kind of most common type of combination of all these terms is a photoautotroph. This is what we typically think of with a photosynthetic organism like plants or algae or some types of bacteria. Photo is they get their energy from light. They're doing photosynthesis, cyclic or non-cyclic photophosphorylation. They're using light to drive their electron transport chain to make ATP. That's their energy source. Auto is they have a pathway that they can take carbon dioxide out of the atmosphere and fix it into glucose or other kind of sugar molecules. So photoautotroph, all right? So these are the two most common organisms, the two most common and probably the most studied kind of chemical energy requirement combinations. Well, let's take a look at some of the weirdos, the ones that are kind of mixing and matching these ones. So let's take chemoautotroph. So a chemoautotroph is an organism that gets their energy from breaking down molecules. So they strip electrons away from organisms to drive their electron transport chain, but then they can produce their own sugars via carbon dioxide. So there's just a few types of bacteria and archaea that fit into that category. And then lastly, we have photoheterotrophs. That was kind of dark. So again, this is the other kind of weird mix and match where photo is that they get their energy from light. The light drives their electron transport chain to make their ATP synthase work. Hetero, though, they have to take in nutrients to get their carbon to make their carbon molecules. They're not getting it from carbon dioxide. So again, it's a smaller group of certain types of bacteria and some archaea. So of the combinations that we have here, these are the two most common, but do know there are some weirdos that kind of mix and match and do things slightly differently. Okay, so in the notes, you have the list of all of these photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Okay, so that's your chemical and energy. But before we get done, I wanted to mention a couple other terms that are on this part of the notes. We have what are called organotrophs. So organisms that use these organic molecules as a source of those electrons, like we would be a chemoheteroorganotroph, if we wanted to fit that in there, is that the chemicals that provide our energy, right, the chemos, they also provide the electrons. So that's what we saw back in chapter 5. Glucose, which is our organic molecule, provided the electrons that we stripped away into the NADH and FADH2. It was an organic molecule, so that gives it organotroph. It's where those electrons come from. Now, there are other organisms that get their electrons from inorganic things like minerals and salts and iron and stuff like that, and so they would be called a lithotroph. So lithotrophs get their electrons to drive their electron transport chain from inorganic sources. So you could have any combination of these things like chemoautolithotroph, photoheteroorganotroph, photoautolithotroph. So it's the organo and litho is just another kind of pinpoints it a little bit more as to where the electrons come from that are being energized by either the chemicals that they're coming from or the light that they're being used, that's being used. Most organisms are organotrophs. There's just a few lithotrophs out there. Okay, so those are the sources or the requirements for chemical requirements. That's your carbon source needed to build more organic molecules. And energy, how do you excite those electrons to drive your electron transport chain to make your ATP? All right, so that's our first little mini lesson from chapter 6, and we will move on to oxygen requirements. See you next time.